everything. But yeah, so I'm a graduate student in the Department of Microbiology and in conjunction with the Hawaii Institute of Marine Biology. And I do work all about coral disease and the microbes related to coral disease. So corals are animals, and animals can succumb to a variety of different diseases, including corals. And this picture on the front slide here is what one type of coral disease may look like. The coral that's red and kind of brown color, that's what it's supposed to look like. And sometimes when they get diseased, they lose the tissue, exposing the skeleton underneath. And this can be a really bad thing for reefs if there is too much disease, because corals are very precious resources, and these diseases can take out stretches of coral reefs in a very short amount of time, as opposed to the tens, 20, decades, hundreds of years it takes for corals to get kind of to the size that they are. So it's really important to try to understand like what is happening to these corals when they get diseased, so we can maybe try to do something about it and keep our reefs healthy and intact. And this is particularly relevant in Hawaii, where it seems like coral disease is getting worse and worse. One of the first outbreaks of coral disease was reported in 2003 in the northwestern Hawaiian Islands at a place called French Frigate Shoals. Since before then, there really hadn't been many reports of coral disease at all in the Hawaiian Islands. But recently, we've had many outbreaks: 2009, 2010, 2011, 2012, 2013, 2014, and luckily we have yet to see one for this year. So this may be showing that across the Hawaiian Islands, coral diseases are increasing over time. Oh, that should be 14. Yeah, so the frequency of outbreaks, so large numbers of corals getting disease at once, is increasing over time. We also have some evidence to suggest that maybe the severity of the disease outbreaks is also increasing over time. So this is an example in Kaneohe Bay, where in 2010 we had a disease outbreak, and there was a multi-agency response. So DLNR's Division of Aquatic Resources, Manoa, USGS, we all responded to this very big outbreak in Kaneohe Bay. And we counted close to 350 coral colonies with signs of disease in the regions of Kaneohe Bay that are highlighted in yellow here. So we swam across all of this reef and counted every coral that we came across that had disease. All right, so less than 400, but pretty large number of corals, 2010. When a similar disease outbreak was reported in 2012, we did the very same thing. This multi-agency response went out and swam over all of these reefs marked in yellow. But now, this time, we have over 1,000 corals that were documented having coral disease. That's triple the amount of destruction and death happening on the second outbreak. So this is kind of alarming. We really need to figure out what's happening here. Why are we getting more coral disease? Why is it getting worse? And if you notice these numbers, these correspond to different regions of Kaneohe Bay. So SB being the southern region, central, and north. And we see that most of the disease is occurring in the southern regions of Kaneohe Bay for these disease outbreaks. So the name of this disease, we call it Montipora white syndrome. So what that means is that it affects the coral Montipora, which is kind of commonly known as the Hawaiian rice coral. And white syndrome means that it's a tissue loss disease. The coral is losing its tissue, similar if you had like a flesh-eating bacteria in a human. It's just dissolving the tissue away, and all you see left is that skeleton that the coral secretes, and the skeleton is white. So this is why we call it a white syndrome. So the research that I'm doing for Sea Grant is really looking at what makes the coral susceptible to diseases. Come on in.
See, there's still a few more choices.
Yeah. Hey. All right, I'll really quickly recap. My name is Amanda. I'm a graduate student at Manoa, and I do work on coral disease. Coral diseases are a concern here in Hawaii, and if we want to protect our coral reefs, we need to understand disease. I study a disease called Montipora white syndrome, which affects the Hawaiian rice coral. It can kill coral rather quickly, so we really want to figure out what's going on here. And then my research is centered around risk factors. So like the title of my talk, what makes a coral susceptible to disease? And there's two kind of facets that I'm coming about this. How are corals becoming exposed to pathogens? That is one really easy way that as humans, when we study human disease, we try to figure out, how is a virus being transmitted from person to person? Where do we pick up the bacteria that cause different diseases? So that's an obvious first step to do for coral diseases as well. So I will summarize kind of the past year and a half of research that I've done searching for coral pathogens. And then for the second part of my talk, I'm going to talk about what makes a coral kind of vulnerable to infection with specific bacteria. And that has to do with some of the environmental stresses that happen in Kaneohe Bay. All right, so I'm first going to talk about this first one. My lab at UH Manoa, the Sean Callahan, Greta Abbey kind of research lab, we have identified 3 bacteria that we can prove cause disease in this coral. So I kind of have a shorthand for them, and I have a really cool methodology of trying to screen them from environmental sources. So I'm not really going to go into detail into this unless you guys want to know later, but I have a way with culture methods and a way with kind of molecular methods to confirm the presence of any one of 3 pathogens in any sample that I collect. All right. So 2 of our pathogens are Vibrios. You might have heard that before. They're common human pathogens as well. And one of our pathogens is called Pseudo-Ultramonas. But if you just kind of look at the color scheme here, that'll be a good way to look at it. So for the past year and a half, I've taken samples of seawater in Kaneohe Bay, stream water from streams that flow into Kaneohe Bay, and various reef-associated organisms between the coral itself, kind of sessile invertebrates like feather duster worms that brush up against coral, as well as animals that interact with coral. So this is a little goby fish. He can actually touch coral and sit on them and use the coral as its home. This is similar with the shrimp. And then butterfly fish, some species of butterfly fish here actually eat coral, so as a mechanism of transfer and a wound. So I have screened many individuals and many samples from all of these different categories of things. And I haven't found a pathogen in every single sample, but in every different type of sample, I have found at least one of our 3 bacteria that we know causes disease in the rice coral. So I can always find our Pseudo-Ultramonas in seawater all the time, and not just in Kaneohe Bay, outside of Kaneohe Bay too. I have found one of our Vibrios in stream water once, and then in all of these organisms at least 2 or 3 times from just 10 samplings. So out of 30% of the time, I find one of our pathogens on an animal. So this is, who knows how widespread this particular bacterium is. There's hundreds of animals that could sample to look for them. So that is kind of a summary of that first question that I was getting at. How are corals becoming exposed to pathogens? Well, they're probably being exposed to pathogens by the water that's going across them from the animals that are interacting with them. And as you can see, this is the coral itself. The coral that gets sick harbors its own pathogen. This is very similar to us, where we harbor our own pathogens on our skin, like Staphylococcus, and we only get disease from them if we get a really bad cut or for immune compromise, right? So that kind of leads me to my next question. Since these pathogens seem to be widespread in the environment, what makes a coral vulnerable to infection? So the hypothesis being a nice healthy coral can keep the pathogens at bay like we do when we're nice and healthy. So when are they not healthy? And that's a whole slew of ideas that you can start going down. But I wanted to look at this question about Kaneohe Bay. So when I told you before during the outbreaks, a lot of the coral disease was happening down here in the southern region of the bay. So I wanted to ask, okay, what's different about these 3 regions of the bay such that we get so much disease in the southern portion? Well, one really obvious, even from this picture, is the amount of urban development in the southern region of the bay, and that can have its own slew of different stressors to coral reefs. One has to do with the residence time of seawater. So in the northern and central part of the bay, seawater in this area easily gets flushed out from tides as well as from water flow within this channel and this channel. So reported residence time, 3 days. We followed a particle of water that entered that region of the bay about 3 days before it finally left. As opposed to in the southern bay, reported residence time is 13 days. So less water motion down here is basically the take-home message there. And then the last one that I really thought about was freshwater input. Corals obviously live in seawater and they're adapted to a certain salinity. In Kaneohe Bay, that's around 35 parts per thousand salt concentration. But as you might have noticed, the last couple of weeks we've gotten a lot of rain, and a lot of that rain just goes straight out into the ocean. It doesn't get kind of absorbed as much as into the groundwater as much as other places. So freshwater input can actually be a really big stressor in kind of areas around Hawaii and especially in Kaneohe Bay. And with that longer residence time in South Bay, we might have a larger freshwater kind of stress. Right? So I'm thinking that this might be something that's affecting corals. A longer residence time with higher freshwater input in the southern region of the bay could be distressing out our corals in a way that allows pathogens to infect them. All right, so I needed to put this to the test. So what I did is I set up this really complicated experiment, and I'm hypothesizing that a salinity stress on our kind of rice coral, Montipora, can either affect a couple of things. I'm looking for time it takes for our bacteria to cause infection in the laboratory, or the amount of bacteria that's required to cause infection. Normally, when we do these lab experiments, we have to put a lot of bacteria on a healthy coral to get it to show signs of disease. 10 to the 8th bacteria per milliliter of water. And that's about 1 billion bacteria per milliliter of water. That's a lot of bacteria. Anything less than that, we don't really see infection in the laboratory. That's not really realistic to what's in the natural environment. So finding things that allow a lower doses of bacteria to cause infection is kind of would be relevant to know about. So I set up this experiment inside the laboratory where I have some corals in these aquaria, and these are my control corals, and they received no stress. They're nice and healthy. They've got 35 parts per thousand, the normal concentration of seawater. And then on kind of this part of my experiment, I have corals that were treated with a stress. So I brought the corals down to 20 parts per thousand, which is kind of the dip in salinity during heavy rain events. And I kept them there at 24 hours and I brought them back up. And at the end of that stress, I inoculated all of these corals with a pathogen, one of 2 pathogens. So first I tried one of our Vibrios that normally, like I said, takes lots of bacteria to cause infection, and it usually takes 5 days for it to cause infection. Okay? So I inoculated corals with the normal amounts, 10 to the 8th, so 1 billion bacteria per milliliter. These corals got 2 orders of magnitude less, so 10 to the 6th, which is about 10 million bacteria per milliliter. These corals got another 2 orders of magnitude less, so 10 to the 4th, or about 100,000 bacteria per milliliter. And then these didn't get any bacteria at all. So the first experiment I did with this pathogen called Vibrio corlyticus, and we call it OCN008. And this is a graph of how well they did, who survived after 30 days. So normally, the control, so unstressed coral with lots of bacteria, maybe 40% of them survived, but they died rather quickly. That's what this graph is showing. If I stress the corals out with a low salinity at the same amount of bacteria, they kind of follow the same pattern. So in this instance, stressing them out with a lower salinity didn't change the time to infection with our normal 10 to the 8th dosage. However, when I lowered the dosage that normally 100% of them survive if not stressed. When I stress them out, I do start to see infection. So this is really exciting. Normally in the lab, we can never get them to cause infection at this dosage with healthy kind of unstressed corals. But as soon as the salinity is dropped, even for just 24 hours, over 50% of them are succumbed to this bacterium. So that means salinity stress allows fewer bacteria to cause the same infection. All right. Next, I did the same experiment, which is actually an ongoing experiment. I've only done 15 days so far, so this is what this graph is showing. This pathogen, let me go back. This pathogen, Vibrio owensii, that we call OCN002, at the same kind of high dosage, 10 to the 7th, 10 to the 8th, it normally takes a long time to cause infection, 28 days on average. So that was, 28 days is what I expect if I don't stress the corals out at all. So far, even the unstressed corals with lots of bacteria haven't shown signs of infection yet, but it's only been 15 days. But I've already started seeing infection on the stressed corals at that same dosage. We've never seen infection at 5 days with this particular pathogen on a normally healthy coral. So the salinity stress did change the time to infection with a normal dosage. And I'm waiting to see if it also affects the dosage necessary to cause infection. So that kind of what this means now is when corals are stressed out with a rain event where the salinity drops, they're more prone to getting infection. Kind of like if we go out into a cold, rainy day and get chilled, we're pretty prone to getting a cold virus or a flu virus. Kind of similar concept. So during heavy rain events, we might want to start keeping our eyes out for coral disease, at least in Kaneohe Bay. So what does this mean for Kaneohe Bay reefs? Kind of touched on some of these. It looks like this particular coral is constantly exposed to its own pathogens, either because it harbors them themselves or it interacts with animals that have the pathogen. So limiting exposure of pathogens is probably not going to be a feasible idea. If it was only on one organism, we could talk about whether it's worth keeping the populations of that organism low, but it seems like it's everywhere. So we can't avoid the flu virus, it's everywhere, so we kind of have to come up with other options to keep us healthy from the flu. Similar idea here. But it looks like these other risk factors, for example, the salinity stress that I tested, allow for easier infection. So there's a couple ideas that you can think about to keep corals more healthy during rain events. You can try to limit the amount of fresh water that gets kind of thrown into the bay by the way you interact and use our streams. But of course, there could be a bunch of other stressors that have yet to be tested that might also allow easier infection. So there's a lot more work to be done. It's pretty exciting that we're starting to get an idea about how these pathogens work, where they're going, and how they're interacting with coral. So this is a very big project that I can't do unless I have a lot of help. And I want to thank everyone in my lab, everyone of my volunteers who's helped me, and I especially want to thank Sea Grant, who's my funder and big supporter, and it gives me the opportunity to talk to my research with you guys. And like I said, how now that we know when coral might be susceptible to disease, we really need people out in the water to be watching the corals so we can find coral disease when it happens. And there's a program in place to help us do that. It's called Eyes of the Reef. It's a network of people who love the ocean and are in the ocean. And we train you to recognize coral disease and other threats, but I'm most interested in coral disease, so that when you are out on your reef and you see it, you know where to report it, and you know what actions will be done when you report it. So I'm a trainer. I'm a big part of this program. So if you guys have any questions about my research or about this particular program, please ask me any questions, or if you want to go to our Facebook page or our website, which is eorhawaii.org, you can find tons of information, and I hope you guys get involved. So I'll take any questions, any comments, and we can have a discussion. So thanks. Yes.
Any questions?
I have a question. question.
Yeah.
I say I had one, but I forgot.
Oh, okay. Well, maybe you'll remember it.
When we were doing the various things.
Obviously, it's been raining and there's been runoff for thousands of years. Is your theory that it's different because of construction and the development that more fresh water is?
That is certainly a hypothesis. The streams, especially in the southern region, are quite different than they would normally be without all the urban development. So, for example, Kaneohe Stream is heavily channelized through most of the region before it hits the bay. And that allows water to run really fast instead of kind of settling out and being absorbed by local plant and the ground. So that is a working hypothesis, and kind of doing this northern versus southern region kind of comparison kind of help us get at that question.
Do you know the fact that the two pathogens are pathogenic?
To this coral?
To the coral?
Yes, so the way we go about kind of proving this is we take bacteria that are associated with a diseased coral. Actually, we use the medical model and we fulfill what's called Koch's postulates, which is a series of experiments to prove disease causation in the medical field. And we have done this with these two corals. So we take bacterium from a diseased coral, we isolate it out into individual bacteria. We then put them back on this healthy coral, and whatever bacteria causes disease signs, we see if we can pull that same bacterium back from an experimentally diseased coral. And that's part of the process of proving Koch's postulates. You can't just put any bacteria. We try a whole range of bacteria. Most of them do not cause disease signs. So it's very specific to what we are calling our established pathogens. Yes?
Okay, so given a particular coral and let's say the infection is with a single coral,
Okay.
what's the transmission? How does it transmit to the other coral?
In some cases, disease can transmit if corals are touching each other. So kind of the lesion and the kind of necrosis of the tissue spreads from one spot on the coral, and it spreads to other parts of the colony. And if two corals are touching, it can sometimes transmit from one coral to the other. Transmission is an ongoing kind of series of questions that we're investigating that we're not 100% sure on. So if you keep a coral in an aquaria that's diseased and keep a healthy one not touching, even just in an aquaria with, little water motion or little mixing and dilution, the healthy one is not going to get sick. So there's probably not a lot of waterborne transmission, but we don't know. Like, we haven't done a whole lot of experiments about transmission just yet.
Yeah, have you studied why it is and it takes so much of the pathogen in the laboratory to diseases when you don't have anything like that exposure?
Right. Some of my lab members are looking at how does the pathogen cause disease. Like, what are the virulence kind of factors associated with them? One of them, the bacterium that causes disease very quickly, the 5 days, the Vibrio paralyticus, it has its own antibiotic. So one of the kind of theories is that it produces its own antibiotic, kills the other bacteria that are normally on a coral that protect it, and then is able to take over. But you need kind of amount of time for that pathogen to get into sufficient numbers before it can make that antibiotic. So it needs a period of time potentially to kind of get populous enough to start being kind of pathogen. I guess why we had to put so much on it in the lab, it could just be a condition. The corals are normally quite able to resist it until you get to a certain number of bacteria, and then it just succumbs. So that's why I'm looking for situations that allow for less bacteria to cause disease. Like the
munching on it.
Like somebody munching on it, or the salinity stress, or sedimentation stress is another one that I will be looking into in the next coming months. There's so many variables that you can look at. I can only do so many. Yep.
You're not looking at heat stress.
I am not looking at heat stress, but obviously heat stress and coral bleaching is often associated with coral disease. So there's been a lot of kind of work on bleaching and coral disease, but we don't see outbreaks of this particular disease during the summertime. There have always followed heavy rain events. in the winter time during the rainy season, which is why I focused on salinity stress and sedimentation. Yeah.
Inside of the lake producing salinity. So is it because of the urban, or is it just the residential that's causing the majority of the problem?
That I can't answer from this research, but it's kind of both things, right? The longer residence time and the greater amount of fresh water kind of maybe be working in synergy. So it would be good to test this in other places that don't maybe have that dichotomy to tease apart.
And
Yeah.
also the urban, there's so much, you don't know what's going on.
Right.
Besides, like just recently, the flooding, and you have city, sewer and stuff. That
Mhm.
really deal with it.
Yeah, that's a problem definitely too.
And the bacteria that you tested, those two, they're just natural pathogens. They are meant to do.
They're not associated with human flora. No.
Okay. So they're just natural pathogens.
Yeah, they're marine bacteria. Any questions about eyes of the reef? I do not have materials to give to you. Maybe. You guys might have some, I'm not sure.
I'll look at the office, so I'll just go right down there and grab it.
But our website is a really good resource to kind of look at who we are, and you can actually go in and do some of the training yourself online. So
You said 3 of something. One is by culture, one's for molecular.
for my identifying my pathogens, we have 3 pathogens, and I use molecular methods and culture techniques to identify them. So, yeah. So I have a little dichotomous tree. I grow bacteria. There's a type of media that allows for only Vibrios to grow. So if it grows here, I know it's one of these guys and not that guy. Like I said, one of them makes an antibiotic, and that's what that looks like on media. This guy is preventing the growth of all this down here. So if it has an antibiotic, it's probably this guy and not that guy. It's the way my methodology kind of works. So I can talk about that more if you guys really want to know about it, but it gets rather technical. So.
I figured out why you have to have so much milk. It's because of the fact that you're trying to make one particular small sample diseased rather than taking a huge number and having sort of randomness.
Well, we definitely don't want to be growing bacteria and dumping it on, like,
large number.
right, on the reef.
So the percentage-wise, you might get 1 out of 1,000, and that wouldn't be good enough for the laboratory.
Yeah, I mean, the corals are quite adept at protecting themselves. So the corals have a mucus layer like the inside of our mouths that are constantly sloughing off bacteria. So even if you're putting a lot of bacteria on, it's probable that a lot of them are just being sloughed back off into the water. So only a small amount are able to attach, stay there, and then cause the infection process. And it can just be a game of probability, where the more bacteria you put on, the more likely you are to get.
So what's something that you see if you're in a certain area, you said that then you know what will happen after you report. Is it what your research would do afterwards, or should you go out there your research and kind of like for animals doing study, same?
So when we get reports from Eyes of the Reef, one of them goes to kind of the coordinator, who's a researcher, Greta Abbey. But the other one, they also go straight to the Division of Aquatic Resources. So depending on the report and the severity of the reports, either we will send someone to just verify what you saw, or we'll do that anyway. But or we will enact this whole process that the state has. It's called the Rapid Response Contingency Plan. We do this for the bleaching events, for the disease outbreaks, and that's part of the multi-agency response, where we document, take samples for research, and then in some cases look at probable treatment methods. So there's a big downstream process that happens if a report of a certain type comes in.